Ductal carcinoma is an uncontrolled growth of malignant epithelial cells in the lining of the mammary glands of the breast. In women, the breasts are modified glands that produce and secrete milk and consist of three main parts. The glandular tissue of the breast is the milk-producing part, which consists of both ducts and lobules. In females, this tissue grows in response to sex hormones such as estrogen and progesterone. The stroma consists of connective and adipose tissue and makes up 90% of the overall breast. The lymphatic vessels collect and drain waste from the breast.
The mammary duct consists of the lumen, the basement membrane, the myoepithelium, and the epithelium. When hormones such as estrogen and progesterone are present in the body, they bind to the receptors and stimulate cell division and proliferation of the epithelium in the mammary glands. When these hormone levels drop, cell division and proliferation should discontinue, cell death should occur, and the dead cells should be removed from the lumen. Certain genes have been identified in regulating both cell growth and programmed cell death. The erythroblastic oncogene B, located on chromosome 17, codes for the human epidermal growth factor receptor protein, otherwise known as the HER2, which is embedded in the epithelial cell membranes and binds with growth factors to stimulate cell growth, division, and repair. The breast cancer 1 and 2 genes, located on chromosomes 17 and 13 respectively, code for tumor-suppressing proteins. These proteins help to repair damaged DNA and ensure cells divide at a controlled and moderate rate. They also stimulate apoptosis when cells are proliferating too fast or when there are too many cells present.
Ductal carcinoma in situ is an abnormal proliferation of epithelial cells that line the ducts of the mammary glands. The abnormal proliferation begins when a dividing epithelial cell mutates, resulting in a malignant tumor cell. The exact cause of this mutation is not completely understood, but there are a number of theories suggesting potential causes. One of these theories being an increased exposure to hormones such as estrogen and progesterone. It's believed that increasing the amount of times the cells will divide and proliferate increases the risk of mutations. Another theory is related to genetic mutations, whether inherited or acquired. An inherited mutation in the BRCA gene can lead to decreased synthesis of tumor-suppressing proteins, decreased DNA repair in mutated cells, unregulated cell growth, and decreased cellular apoptosis. An acquired mutation in the ERB gene can lead to an overexpression of HER2 protein receptors in epithelial cell membranes. This means an excess of growth factors are able to bind to one cell and cause excessive cell growth, division, and proliferation. In DCIS, the tumor cells have invaded the ductal lumen but have not broken through the basement membrane.
Invasive ductal carcinoma is when the in situ carcinoma has penetrated through the myoepithelium and basement membrane and has begun proliferating into the stroma.
Let's take a closer look at the layers of the mammary duct and what happens when homeostasis is disrupted. The purple layer represents the basement membrane, the red cell layer represents the myoepithelium, the innermost layer of cells represents the epithelium, and the malignant epithelial cells are inside the lumen. The process of invasion begins when trauma occurs to the myoepithelial cells, causing them to break open and spill their contents into the ductal lumen. These contents are considered chemoattractants, which stimulate lymphatic cells from surrounding lymphatic vessels to enter the lumen. Once in the lumen, the lymphatic cells become activated and release a number of enzymes, one of these being protease, an enzyme which aids in the breakdown of proteins. In the ductal lumen, the protease breaks down the proteins of the basement membrane, which results in gaps that lead out into the lumen in the stroma. The tumor cells present in the ductal lumen are now able to penetrate through the basement membrane and proliferate into the surrounding stroma. Once in the stroma, the tumor cells can metastasize very quickly into surrounding tissues and structures such as muscles, lymphatic vessels, bones, or vital organs.
As discussed, there are a number of risk factors associated with breast cancer. One of the major risks is being female. and another being prolonged or increased exposure to hormones such as estrogen and progesterone. Prolonged or increased exposure to hormones may be associated with early menarche, late menopause, few pregnancies, or hormone replacement therapy. One of the other main risk factors is genetics, which can be due to either inherited or acquired mutations. Changes in breast tissue can often occur in response to hormones, and many women may find their breasts feel lumpy due to the fibrous tissue that makes up the stroma. But a lump that feels harder or different from the surrounding breast tissue should be considered abnormal and should be further examined. Other changes in the breast or nipple can include dimpling of the skin due to obstructions in lymphatic drainage, nipple inversion due to shortening of mammary ducts, rash, redness, swelling due to inflammation, discharge due to obstructions. Pain, discomfort, or tenderness in any part of the breast or underarm region is a result of inflammation, which may or may not be felt.
This chart shows the prevalence of breast cancer in Canadian women in 2015. Less than 1% of diagnoses occurred in women between the ages of 0 to 29, and greater than 50% of diagnoses occurred in women between the ages of 50 to 69. Another statistic determined that 1 in 8 Canadian women will be diagnosed with breast cancer in their lifetime. As of 2018, breast cancer is the most common cancer in women. Normal homeostasis is disrupted when an epithelial cell mutates, resulting in a malignant tumor cell, which will divide and proliferate at uncontrolled rates, resulting in ductal carcinoma in situ. The cell membranes of the cells in the mammary duct are damaged by the tumor cells, resulting in proliferation of malignant cells into the stroma, where they can then easily metastasize throughout the body. The HER2 receptor protein can be overexpressed in the malignant tumor cells. As well, when BRCA genes are mutated, the tumor-suppressing proteins they synthesize are no longer effective.